It's really now my great pleasure and great honor to introduce the first speaker of the workshop, Professor Ingrid Daubechies. Professor Daubechies currently holds the title of James Duke Professor of Mathematics and Electric and Computer Engineering at Duke University. As I'm sure most of us know that Ingrid has made many fundamental and transformative contributions to mathematical methods for the analysis of signals, images, and data. Her work is a permanent contribution not only to mathematics, but also to science and engineering. She's especially known for her groundbreaking work on wavelet theory that has catalyzed multiple innovations and that has transformed the numerical treatment of images and the signal processing used in technologies ranging from medical images and wireless communication. Yeah, has won most of the major awards in mathematics, science, and engineering, which is really certainly 2 minutes list here. But let me just briefly mention that Professor Ingrid is a member of the National Academy of Sciences, National Academy of Engineering, and American Academy of Arts and Science, and also a MacArthur Fellow. It is again really a great honor for us to have Professor Daubechies now talk about her recent work on no-dimensional manifolds in high-dimensional data sets. Please, Ingrid.
Okay. Great. Well, thank you very much for this very nice introduction. And I have to apologize a little bit to the attendants that although I have done some recent work related to deep neural nets, this talk will not be about deep neural nets, but not because I'm against them. As I said, I share completely Jin Chao's motivation that we want to understand deep neural nets. I don't view them as alchemy. I view that we are, with respect of what neural nets can do, a little bit where people were in the 19th century when in understanding electromagnetism. I mean, we observe things and they're peculiar and we haven't understood the whole hanging together. And the work that is being done by mathematicians, like what Jin Chao quoted and others are doing, is helping us getting there. But I think we still have to understand more to see why they are so peculiarly successful at the things they are successful at. Because some of those were hard problems that people have worked very hard at for many years, and we didn't manage to crack them as well as deep neural nets have done. But this talk will not be about that, but I'll tell you at the end why not, and how we hope to change that. Okay, so well, this is the picture in the back of my mind. If I have something like this, which is a simple, really a flat thing, but folded and sitting in 3D, then if you have sampled points from it, you'd like to discover the geometry of the blue thing, even though it's given to you in 3D. Now, of course, this is easy because we can see it. But in general, we'll have things that have structure and that are in much higher dimensions, and we want to find out that structure. And the method that I like to use for that is diffusion maps. Now, it has its limitations. It means that you must have sufficiently closely spaced and data and representative, sufficiently sampled to be representative at least of the portion of the manifold that you want to discover. And you are assuming it is organized like a manifold. But if that is the case, and there are data sets for which one can really argue that that is the case, then you want from these samples to infer the geometry. And if you go back to the example, then it's clear that small distances when distances are small between your sample points, they are very close to the geodesic distances. But when they're large, like distances between the point here, where my mouse is now, or where it's now, these are in R3, in the ambient space, much less reliable. So that's why we will look at neighborhoods of points, and these neighborhoods there will be many small neighborhoods and they overlap. Within each neighborhood, you can use distances, and as a result, you can use a way of looking at the geometry. And that for me is a little bit similar to how you can see that these animals are surfaces, even though what you see are all these little sequins, which are little flat discs covering it. But together, they give you a very clear impression of the surface, even though you can't see any of the underlying thing on which they have been sewn. So in each of these neighborhoods, I can use the distances to define diffusion. And then I can use the points that are in overlaps of the neighborhoods to link from one to the next. And so I can link several of these paths within each neighborhood together. And that makes it then possible to actually think of diffusion paths that link points that are far apart, that are not within the same small neighborhood. And that's really what you do with diffusion methods. So my view, I mean, the manipulations I do here are exactly those, are exactly those that are done by people who do diffusion maps, à la Bilkin-Yogi, or as Kaufman and Lafont do. But my interpretation may be slightly different. So my interpretation is that I'm assuming that typically the distances I have, the ambient space I have is an Rn space. And so in, or D space, with D very, very, very large. And then the diffusion in Rn is really given always by the kernel e to the minus distance between points squared over 2 tau. The normalization factor depends on the dimension. But let's not think about normalization for the moment, because I'll have to tinker with that anyway. So depending, regardless of the dimension in which I live, I always have that same kernel. So let's take that ambient kernel, just like I would diffuse on the little sequence, but I will only diffuse between data points and neighboring data point. I mean, nearby data points. Tau will be fairly small, so that this exponent here becomes becomes way too large when the distance between points is large, and so then I put things to 0. So I look at this matrix, which is going to be a sparse matrix, and I want to use it to define a numerical approximation to what I hope will be the diffusion on this manifold. So that numerical approximation better have the properties of a stochastic matrix as the operator that is the diffusion operator, which I use for, which is generated by the Laplace-Beltrami operator on a manifold also does. And I'm using here, I mean, I'm measuring my distances in Rn, but I'm using that I hope to have a manifold, and that the distances in Rn are the same as the distances in that tangent space. And I have small distances, so the distances I measure in Rn are very close to the distances that I would get from the exponential map or on the tangent space. And so I do this confusion, but which is mathematically justified, between the distance in the ambient space and the small distances on the manifold. So I have, I believe, a kernel that's very close to the right kernel, because I'm at small distances, and I normalize it because I didn't know the right normalization factor to put there anyway. And I have to take into account that my sampling is not regular and so on. And also some points may have more near neighbors than others. So I do that by taking the sum over all the entries, which are not which link one point to others, and putting that as a diagonal matrix and defining this product matrix, which now will be a stochastic matrix. Okay, so in this method, people often say that picking tau can be hard. You don't, so it's more an art than a science. And I have found that, at least in the applications in which we work, taking this idea of diffusion seriously actually helps. So the idea is we are building an approximation to the diffusion on the manifold. The true diffusion operator will have a semi-group property, namely that if you, and this is has been studied by my student Shan, and where the paper is being written up as we speak, a true diffusion, if you have the Laplace-Beltrami operator on the functions on the manifold, will have the property that the product rule, that if you diffuse by time t and time s, that is the same as diffusing over time t plus s. So what we do is, grosso modo, we pick tau so that we pick the largest possible tau. You don't want to pick tau too small. If you pick tau too small, then you have no entries because all the exponents are too big and it becomes meaningless. So you want to pick tau as large as you can afford to, but you have to still have a diffusion. So we pick tau so that the square of the stochastic approximation matrix is still close to the matrix that you would obtain for 2 tau. And that has given us good results in practice. Okay, so once you do that, you do a spectral decomposition. So you find eigenvectors, actually not of this, well, you find right eigenvectors of this, which is the same as eigenvectors of the symmetric matrix D minus a half, W D minus a half. And then once you have these eigenvectors and their eigenvalues, you can immediately think conceptually of looking at diffusion for larger times. All you do is you take the corresponding power of the eigenvalues. And that's really, for me, what is behind this defining of a spectral distance that is done by Lafont and Koifman, for instance, where they identify the coordinates you get from the eigenvectors with a new parameterization of the manifold, and they take powers of the diffusion operator. Okay, so when you do that, so here's an example where we have 50 points, which are really 5 species of 10 points of different 5 groups. And the distances, when we did multidimensional scaling on the distances we had obtained, and I'll come back to those distances in a bit, then this is how the distances distributed in the best possible 3-dimensional projection. And so you see this points of the same sort do cluster a bit together, but it's not a great clustering. Now, exactly those same data in multi-dimensions, if we apply the diffusion map to it and then do the multidimensional scaling on what we obtain, we get the clustering on the right. So you see that you do get a much better geometric interpretation in this case for this manifold and diffusion picture. Okay, so that's how we got this picture. Now, what is this data set I'm talking about? This data set is really that is a data set that came from biologists. Jukka Jörnvall is the biologist with whom I started talking. He's a very eminent paleontologist and evolutionary anthropologist in Finland. His postdoc at that time, Doug Boyer, is in the meantime, this is we started talking about 12 years ago. So Doug Boyer is now by happenstance, completely serendipitously, became a professor at Duke University, and so it's very easy for us to continue working together. And we have worked with lots of other young people. I mean, so you see, Jaron Lipman was the first one of my collaborators. He was a postdoc in Princeton when we started on this. In the meantime, he's become a full professor at Weizmann Institute, and some of his students, like Shahar Kowalski and Nadav Dim, are now working with me. And Shahar just got a tenure track position at UNC, and Nadav is returning to Israel next year. Okay, so first we talked with them on a way of quantifying complexity of teeth, and they had a very ad hoc method that they did, in which they imagined the tooth illuminated by different colored lamps, and they would take a snapshot using computer graphics software and then count the changes of color. I said, so what you're looking at is really the change of the normal direction. I said, well, yes, they said. And I said, well, yeah, we can compute that. I mean, that's a quantity that mathematicians like and the computational geometers can compute. And so with year one, we made a program, and the results of that are still something that they find very useful and has become part of a tool. But then once we had established our credentials that way, as people it was worth talking to and people who might give solutions, they talked to us about a much more complex problem, which in a sense is still what we're working on now, except that it has morphed. So originally, they told us, look, the data we acquire are surfaces. Now we immediately asked, as everybody asks as always, why surfaces? I mean, so they look at teeth. So these are evolutionary biologists. So they look at teeth and bones of many different species, and including fossils. I mean, so you can look at all the species that live, but you can also look at fossils. And that's why teeth are important, because for tooth is the last bone to decay. So many more teeth have been preserved than other bones, but they're interested in other bones as well. And it's true that in the fossilization process, a lot of the internal structure of these 3D objects, teeth or bones, are preserved. And so you can have information that is 3D from fossils. But fossils are precious. And so, especially rare fossils. And the collections that own those are not willing to easily lend them. However, Doug Boyer has perfected a method by means of which, whenever he goes anywhere in the world, he looks up all the local collections and he takes surface impressions of these fossils by means of dental resins, the same kind of high precision dental resins that dentists use to make impressions of your teeth to build a mouth guard or to build tooth implants. He uses for these fossils. And so when he comes back, he has beautiful and very detailed impressions of these surfaces of the teeth. I mean, he acquires the data through CT scanning, but it's not of the tooth, but of some resin object that has the right surface. So only the surface is worth recovering. And so that's why we work with surfaces. And so you get a cloud, you clean it up with standard methods, you triangulate it, and then you can render it. And you can put on texture if you want to talk about particular points on it. You can say, oh, the thing that is there next to the 0.5 and so on, as a reference. Okay, so how do they normally work with these? So typically they want to compare teeth. So they have a lot of domain knowledge, and they look at all these teeth, the collection that they want to compare, and they want to test some hypothesis. So they want to measure things and compare and see how things distribute and so on. So they look at the whole collection and they see what this collection has in common. Now they know a lot about teeth, so they know how they grow, they know how things match together in evolution and so on. So every one of those little nodules here, well, on the tooth has a name. Well, not every one of them. Things that you might think or I might think are just as prominent as some others are less important, and others are, but that's the background knowledge. So they put landmarks on these teeth that all of them share, and they mark them appropriately. And then they measure the spatial coordinates of these. And then they use these spatial coordinates as a representation for the shape. And then if you have two shapes, you will take one of the two shapes and put it in as close a correspondence to the first shape as you can, these shape landmarks. And then you sum the squares of the difference of the distances of corresponding pairs. And that gives you the square of the distance, that gives you the square of the procrustes distance. So here's an example of teeth where these landmarks have done. And so you see, I mean, there are things where I might have put a landmark and they didn't. I mean, like the point, the big basin here. And they say, well, that is not as well preserved. It's not as easy to mark as others. And then I say, but there are some other bumps that I think are equally as prominent as things that you do mark. And well, they always have good reasons. So we trust their domain knowledge. They know a lot. They're like our oracle. I mean, so why do they want to move away from this? Well, it's time consuming to do this. It's not very flexible. It's you need a high degree of expertise to do this. And this is especially important because these days they they encourage people who have detailed digital measurements of surfaces to upload them. And so Doug started something called Smorphosaurus, where you can find openly, and for people who'd like to find data sets on which to compute, you can find them there with the author and you can contact them. These are all data sets that have been published, so they are available with permission of the author to work on. And so there are bones, there are teeth, there are shells, there and so on. And the idea is that people could use them for their work, their comparison work, even if they're outside their own specialized domain. If you have a hypothesis that involves measuring shells about which you know nothing, you can find the data there. But you don't have that domain knowledge to do the landmarking, because that's extremely narrow expertise typically. So if you could do that automatically, then that would be great. So they're interested in incorporating such tools. And even among specialists in the same field, sometimes they have controversy. So here again, let me remind you what they do. So if you have your landmark teeth with xj's on one tooth and yj's on another tooth, then you take the about all rigid transformations, you take the one for which the rxj's get closest to the yj's, and you define that summed square distance. And so they wanted us to do that automatically. And for the surfaces here, you see an example of how finely triangulated these surfaces are. Typically, we subsample them because we don't want to work with 10,000 points. We typically go to 1,000 or 500 or so, a few hundred. So we actually find two different distances, one that involved conformal flattening, and one that is a continuous procrustes distance. I actually prefer the conformal flattening distance, but the continuous procrustes turned out to perform better. So let me skip over the explanation of the conformal the first distance, which we called the conformal Wasserstein neighborhood distance, and go to the continuous procrustes distance. Okay, so this is, I skip over this. Okay, so the first thing we had to deal with, if we trying to find a continuous analog of the procrustes distance, is that if it's not landmarked, then we have no correspondences. So we say, well, let's assume a correspondence between the two surfaces. So X lives on surface 1, the C-axis live on surface 2. And the correspondence, we said, let's presume it's area preserving. And of course, biologists said, we don't want it area preserving. It's certainly not going to be area preserving. I said, wait. You are okay with the fact that we would take 1,000, first of all the surfaces are always normalized to 1, because we want to be independent on whether we look at a very big elephant with huge teeth or a smaller elephant with more dainty teeth. I mean, the tooth will look the same, but the surfaces will be different. So we normalize everything to 1 always. Certainly among species, we have even bigger factors if we don't normalize. And then we say, well, you would be okay if, in order to define something, and we don't have landmarks, we would, let's say, put 1,000 points on one surface and 1,000 on the other, and we put them more or less equispaced. They said, yes. Said, and then we have to find a correspondence between them. We'll map injectively and one-to-one these points to each other. I said, yes. Said, well, then we automatically have done something area preserving, because if it's well distributed, then if you take the Voronoi cell among each, then it's going to be about 1,000th of the whole surface. Same on the other tooth. And if we make a one-to-one correspondence, we map 1,000th of a surface to 1,000th of a surface. And I said, but don't worry, we're only going to trust the distances we compute when they're small. And that means the teeth are very similar. And that means there is no big area difference. I mean, they didn't quite get that. It took a couple of years before they understood what we meant there. But okay, so we have here, you have a correspondence. Then you're going to rotate, in this case, it's two-dimensional, so that you make the sum of the distances minimal. And then, of course, other correspondences would give you even a smaller distance. And that's what we define as the continuous procrustes distance. So CP for continuous procrustes. And it also implicitly defines you a mapping, because the best mapping gives you a mapping from one to the other. And so the texture has been put here to get the mapping. And so it does a reasonable job. I mean, even though it is constant area and typically an area is not really going to preserve here, we do a reasonable job. Okay, so we compared the distances with, so here in this diagram, we on the one side, we have a distance diagram as they would have computed with their procrustes distance. And on the other side, we have our continuous procrustes distance. Here we have it for our other candidate. And in this case, the samples have been placed so that similar things are close to each other. So you see that, so especially since we want to trust small distances best, we should be very similar in the diagonal. And you see that that's where our procrustes distance did better than our other candidate. And that's why we took this distance. Here you see an example of how this thing would transport landmarks for bones that are quite different. And we don't do so badly. I mean, if you compare to where experts would have put the landmarks. Okay, so next, so that was the continuous procrustes distance. So now I've explained to you for the group of 50 exemplars, 10 of each genus, how we got to the pictures at the left, the continuous procrustes distance, and the diffusion distance did so much better. But for a while, it actually, when we talked to our biologist collaborators, they were happy with the results, but they were also unhappy really because of the mappings. Some mappings they didn't like, but it turned out they were always mappings between things that were far apart. And we said, but we don't trust those. And we don't use them for computing the diffusion distance. They said, but then how do we find the right mapping for all of them? So we experiment with minimal spanning trees, but of course that's very unstable. We thought of a lot of a number of different alternatives, but then Ting Ran came up with a method that actually not only gives us better mappings, but also increases the accuracy of splitting. So I'll explain to you now how we get to the diagram on the left, which is much better still than the one on the right. So Ting Ran Gao, in his thesis, observed that, yes, we used the local distances, we knitted them together, we got a spectral parameterization, we got a distance, but we threw away a lot of information in that, because we had not only distances between pairs that we could compute, but we also had this beautiful mapping in between. So why throw away that information? They can do much more. If you think about it, then in fact, this unknown manifold of species, which we assume, and you might say, how can you believe that species form a manifold? Well, we look for the moment that species are very closely related. So it is reasonable with the variability within a species and looking at overlap where people wonder, oh, could it be this, could it be that? They're so similar that they actually form one more or less uniform manifold. Okay, I mean, when you look at all the species together, it's probable that we have little manifolds at the end of branches on a tree. But so we're now looking at something that can be considered as a little blanket that covers these little leaves locally. But so every species is one point, but for every species we have a whole bone, a whole surface, a whole tooth. So, and you have a mapping from one to the other. So that's exactly what a fiber bundle is. I mean, so we have that mapping, which is a connection. And in case we have things with a not too distant common ancestor, we have a flat connection because the mapping from A to B and from A to C, since you could, you hope, reduce it to a mapping going from A to D and then to B, or from A to D and then to C, the mapping from B to C should be going from B to D and D to C, so should be exactly what you get by composing B to A to C, because all go via D. So you can try to hope those to find that use the flat connection. Of course, you don't really have the connection. You have a very, very, very noisy version of the connection. So what he did was, so here I explain a little bit about fiber bundles, but you know all of that, examples of fiber bundles, but these are all fiber bundles that are either linear, like a tension bundle, or derived from a linear bundle, the unit disk on the fiber on the tension bundle, which is not our case. We have a non-linear bundle. So a lot of the things that you might expect and know well from standard fiber bundles, we don't have here. But nevertheless, we have a fiber bundle. And so we have triviality. Well, we always have local triviality in a fiber bundle. We expect to have it over larger regions. But that, in a sense, tells you that your connection is going to give you many views of the correspondence. Okay. What you can then do is you can look at this fiber bundle, which means that you take all the points on each tooth and the collection of all the teeth together. So you have now a humongous collection, and you can try to do diffusion on that. And typically, because you don't know much about the manifold of the teeth, you have to introduce two diffusion parameters, because the ratio of the densities with which you sample on your teeth and on your manifold of teeth plays a role. And since you don't know that ratio, you have to experiment. So you can set the one on the surface on the teeth very nicely. You can also set the one on the tooth manifold, but since there your distance might be scaled, you still have a scale factor that you have to experiment with. Okay, so you find then eigenvectors which have labels. The labels are, remember, are always in the eigenvectors. The eigenvectors are defined in all the points in which you have your bundle. And so we have eigenvectors that have certain number of points on the first and so on. Note that this accommodates very well that you might have different numbers of points on different surfaces, which was again something that the biologists were extremely pleased by, because one of their conundrums is, what if they have a fossil that is missing a piece, or any tooth that's missing a piece? But if it's an extant species, they can try to find, if it's not too rare, another one. But for a fossil, you have to take what you find. And if you're missing a piece, then that meant you couldn't use any of these corresponding thing in your whole comparison if you wanted to use the fossil. Now that's no longer the case. Okay, so you find all these eigenvectors, and now you can do many things. First of all, one sanity check is, let's take the three most important eigenvectors, the ones for which the diffusion eigenvalue is the largest non-trivial eigenvalue. And let's just look at what our surfaces correspond to. And lo and behold, it turns out that they map out the same surface in 3D. So that's already an indication that we have captured something common. So we have kind of captured a coarse approximation of a template domain for these two-dimensional of the fiber, if you wish. And it also immediately meant that we had a better way of registering, because before, when you took 1,000 points here and 1,000 points there, it was obvious that you wouldn't have true correspondence, because how likely was it that you had exactly the same point on the red tooth and the blue tooth? Of course, it was 0, the likelihood. So, but what you can now do is you can register to their common domain and a template, and you can say the blue point lies at this kind of convex combination of these red points. And so let's look at that same combination of the red points on the red tooth, and that's where we should map the blue point to, which is great. So that was a beautiful, but then useful things for the biologists came out. For instance, you can, in this huge space of your spectral representation, you can cluster according to one of these spectral distances. And if you do that, then, so this is what you get for 12 clusters. It turned out that the regions that were marked were represented on all teeth. That was not a given. And they actually represented regions that the biologists thought were really structurally corresponding. And they liked very much that they didn't have to have the same area. I mean, for instance, this very dark blue one is very small here, but quite large there. And so they immediately started using this. We tried to hold them. We said, hold your horses, we're going to get better. I said, no, this is already great. We can use it for statistical studies. And one of the theses of a student of Daubechies exploited this kind of correspondence. But you can also say, look, I have a way of encoding the geometry for each tooth, and only after I make a summary of these do I make an average of all of them to get a summary of the geometry on the base manifold. And that is exactly what Tingran used to get this much better correspondence. So these 5 species, first, for a second, the biologists thought, oh my God, maybe you have the evolutionary distance here. No, you don't. I mean, the blue and the purple one are the furthest apart in this group, but they're close together. However, they're both frugivores. So if you see, depending on what they ate, I mean, or whether they ate fruits or leaves or insects, they clustered nicely together, and it's not so strange, given that we were looking at molars, that food preference would have shaped similarity. And okay, so recently with Chen, we realized that we believe that this method can apply much more generally. I mean, here's a completely different application. These are the handwritten digits of the MNIST database. And if you can do an optimal transport map here. And you can even do it between things that are not from the same digit. And you can use that to find a distance. But you also have a mapping now. So you have, again, you can view every one of those images as a fiber. And if you then try to find distances between them without, so just distances and represent, and with some group of 1s and 6s, you get the clustering on the left. But if you do the best possible clustering, once you distinguish use the mappings as well in the way we showed, you actually can really, you don't have anymore this one stray one that it gets close, but it can really be separated from the others. Here's another group where it was separable before, but it's much better separable with our horizontal diffusion distance. Here is the same with more digits. Again, it's not perfect anymore. On the right, you have a stray red one and some turquoise ones, but things become less confused than they were at first. So that's just to say that we can do things, and that we believe we have a paradigm that is more generally applicable, and that's what we're writing up right now. It's clear also for our teeth that we will need to go well beyond this. First of all, there will not be good mappings all the time. I mean, I show here two extremes: primate teeth, molars on the one hand, and then the molars of crab-eater seals, which are just, I mean, you can imagine that it's not going to find a nice diffeomorphism from the teeth on the right to those on the left. But I believe this is because, as I said, we don't expect to have one full manifold. We expect that as things are further far apart in the oops, where am I? Further apart in the tree, they you will have to go back, simplify based on the data, take some features away that evolved and on a branch and that have no counterpart here. So to do simplification, to do a coarse grading, and then if you do sufficient coarse grading, we'll have little manifolds again. And then if you go deeper again, you have to remove some features. So that is something we have to build. We haven't done that yet. I promised you also that I was going to say something about deep learning. Why haven't we applied deep learning for this? Well, the manifold we want to learn is the manifold of the species. And we just don't have sufficient data. I mean, for deep learning, you typically need thousands and thousands and tens of thousands of samples. Now, people do have tens of thousands of samples in museums, but most of them have not been digitized. We typically have to work with a few hundred, and that's not sufficient. But one thing we hope is that, and we do now have enough understanding of these data sets, that we can use this to generate, to populate our manifold in a way that where the people, where the biologist says, this is the first time, the past few months is the first time that we could do this, that they, oh yes, they say, they look like these. When we tried earlier, I said, oh no, no way. But now we can produce surfaces that they could believe were scanned from a real animal. So we can populate our manifold. And so we might be able to do deep learning, but that's a future thing. So I'm going to stop here.
Okay.
Thank you very much.